Hello. So far in Chapter 2, we've been focusing on crosses that Mendel did, and these are crosses that involve experimental organisms. In his case, it was pea plants, but geneticists might use other experimental organisms like fruit flies, etc. Oftentimes, though, we're interested in inheritance patterns in humans, particularly when it comes to trying to understand how certain diseases run in families. Of course, when we're studying human traits, it's not ethical or really possible to control parental crosses, as Mendel did with peas. Rather, what we have to do is to rely on information in family trees that already exist. And these family trees are called pedigrees. Pedigree analysis, it's an approach that's used to determine the pattern of inheritance of traits in people. And so these are the symbols in a pedigree. I won't go through them all. What you might want to do is to pause the slide and take a look at them and then come back. One thing I want to point out is these symbols right here in the middle. So these are symbols that denote presumed heterozygotes, and so they're denoting genotype. Most of the pedigrees in your textbook are not, let me repeat, not like that. Like the ones in the problem sets at the end of chapters are typically, the symbols represent just phenotype. So if it's a recessive disease, the unaffected person could be a heterozygote, or they could be homozygous for the normal allele. So you don't know. Whereas in the pedigree we're going to talk about in a few minutes, it's showing genotype. So that's a confusing point, is that most pedigrees show phenotypes. However, it's also possible for pedigrees to additionally show genotypes. Pedigree analysis is oftentimes used to try to deduce the pattern of genetic diseases. Are they recessive? Are they dominant? Are they X-linked, etc. Genes that play a role in disease may exist as an allele that confers normal wild-type function and a mutant allele that causes disease symptoms. Okay, and it's always bad to use the word normal when we're talking about people, but oftentimes, at least at the molecular level, the word normal means functional. So if an allele is quote-unquote normal, what we tend to mean is it's functional. We're not making a value judgment here about what's normal and what's abnormal. It's more an issue of functional versus non-functional. Diseases that follow a simple Mendelian pattern can be dominant. They can be recessive. And when we get to Chapter 4, we'll talk about different patterns that you might already be familiar with, like X-linked and that sort of thing. And so pedigrees can also be analyzed to deduce that kind of a pattern as well. As far as a recessive pattern of inheritance go, this is just an example. Two normal by normal heterozygous individuals, we mean individuals who are not affected with the disease, on average have 25% of their offspring affected. So again, I don't like to use the word normal. I'm using it here at first so you get a sense of what it means. It would be better to say two unaffected heterozygous individuals, so two individuals who don't have the disease. In the case of recessive disorders, two affected individuals would produce 100% affected offspring, because affected individuals would be homozygotes, so all of their offspring in such a cross would be affected. On the other hand, a dominant pattern predicts an affected individual will have inherited the disease from at least one affected parent. If it's a rare dominant allele, it's probably just one parent. Occasionally, a disease could arise by a new mutation, although that would be pretty rare, but that's another possibility as well. So here's a pedigree showing the disorder cystic fibrosis. Again, this is showing genotypes. So the two individuals here at the top are both heterozygotes. if this was showing phenotypes, these would be open symbols. You wouldn't know that they were heterozygotes. You would just know that they were unaffected. So if the pedigree was just showing phenotypes, open or white symbols would be unaffected, and total black symbols like this one here refers to an affected individual. So now these two heterozygotes also produce, in this case, they produce two affected offspring. Okay, whereas these two individuals here are homozygous for the non-disease causing allele, and all of their offspring are unaffected. Just to end with a little bit about the disease, in case you're interested, cystic fibrosis is a recessive disorder in people. The gene involved encodes a protein called the cystic fibrosis transmembrane conductance regulator. It's actually involved in transporting chloride ions across the plasma membrane, particularly in epithelia, such as the epithelia in a person's lungs or their pancreas, etc. In the case of the mutant allele, it creates an altered CFTR program that's degraded more rapidly, and so people who have this disorder don't transport chloride across their membranes as well as other people do, and that's what causes the disease symptoms. And they're generally abnormalities in the pancreas, intestines, sweat gland, and lungs. And typically, the most prominent symptom is lung infection. So they have very thick mucus because their chloride balance is off. That affects water balance and causes their mucus to be very thick. And that just creates an environment for lots of lung infections. Okay, well, this is just a really short section on pedigree analysis. In the last section of Chapter 2, what we're going to talk about are some statistical methods that can be used to analyze the outcome of crosses.